All right, thank you for coming. Today I'll be talking about TrueAllele forensic solution, making DNA count. And I'd like to thank the organizers for inviting Cybergenetics today. Here's an outline of the talk. I'll begin with a brief introduction, talk about India's needs for forensic DNA, the power of DNA, a seminal case, the Foley case, the first case where modern genotyping was ever used in a courtroom, and then talk about problems with DNA mixtures. As time permits, a few interesting cases, how the TrueAllele system I'll be describing is validated. How does one validate interpreting DNA mixtures? The concept of DNA databases, from some database case examples and their validation, and then how TrueAllele applies to India, and then make a concluding remark or two. So Cybergenetics has been innovating the interpretation of forensic DNA evidence for over 20 years. We were founded 25 years ago. We first solved the PCR stutter problem 25 years ago. We eliminated the United Kingdom DNA database backlog 20 years ago. Most importantly for this talk, over 20 years ago, we solved the DNA mixture interpretation problem with our TrueAllele system. The key concept is to exploit the variation from the PCR experiment to get accurate probabilities. We then worked on the World Trade Center to identify victim remains. We testified in the earliest cases. We did the first exonerations with DNA with probabilistic genotyping. The system has been validated on up to 10 unknown contributors, which makes it the most powerful system in the world. And Cybergenetics has laboratories, but we also has laboratories that use the technology. But we also do consulting work. We've reported in over 1,000 cases, testified almost 500 times. I think I've been to court over 100 times, and it's used in 46 of the 50 states. So here's the problem. For most labs, you have a manual forensic DNA workflow. The genetic analyzer is beautifully automated, but then step by step, you need people. There's automated software, but it's not really automated, is it? A person has to look at a screen and click and make decisions and apply thresholds, discarding data, making choices that are tedious, time-consuming, and often lead to incorrect results. And every step involves people typing, choosing, and the most common result with complex DNA mixtures is inconclusive, meaning that the cases go unsolved and they're never closed. So for all that work, nothing happens. The problem is basically the difference between looking at allele peaks in data, at the surface features, versus forming an informative genotype using Bayesian computing that tries out tens of thousands of different explanations of the data. The old failed allelist method doesn't get answers most of the time, whereas informative genotypes done by modern statistical computing does get answers. I won't go through everything on here in the interest of time, but basically by applying thresholds, as is done in very tedious, time-consuming human review, most of the information for identification in DNA is thrown out by the human analyst. And that's what study after study shows. It's just not there anymore. The answers are either non-existent, that is, there's no answer, it's inconclusive, or the answers give an incorrect statistic, because the tall peaks, the short peaks, the patterns, none of it is left. The older methods use an inclusion statistic, which is all you can use when you've thrown out the heights of the peaks and the low datas. That's called CPI, combined probability of inclusion. Databases that rely on it include CODIS or small pond. The genotypes produced by the informative systems use likelihood ratios that don't have any thresholds. They fully explain the STR data. So TrueAllele was the first such system ever created, and I think the only one for over 10 years. We innovated the notion of how you have no human intervention. It's fully automated. There are no thresholds. No calibration is needed because using Bayesian computing, the TrueAllele system reaches into the data and uses the data to work out parameters like stutter or variation of the DNA. On the left, you see interfaces that are provided for Windows or Macintoshes. On the right is a powerful supercomputers with parallel processing that can solve dozens or hundreds of problems simultaneously, leading to very high throughput for a laboratory, including parallel database matching servers to continuously match genotypes. The result is an automated forensic workflow, whereas you still have the same automated genetic analyzer, but now on the bottom, instead of a manual process of making decisions, clicking on peaks, and taking hours to analyze a sample or ask questions, it's all done by computer. In 1 minute, you can just flow the data files from a genetic analyzer into a TrueAllele system, and questions can be asked automatically about solving the genotyping problems. The results go to databases, the matching is done automatically, and the answers come back from the server. So in speaking a lot with people from India for almost a year, my sense of the needs of India for forensic DNA and scaling up to a country with 1.3 billion people is information, automation, and ease of use. For information, there should not be inconclusive results from DNA mixtures. Whether there's 2 contributors, 5 contributors, or 8 contributors, there should always be information if the data has information, which it almost always does. That will let the labs close their cases. And to do it, it needs to accurately weigh DNA evidence to determine how strongly a suspect is present or not present in the evidence. That's impossible with thresholds and the older manual methods. I think India also needs automation where the computer does all the work. Why try to train and hire thousands of people, new people, for jobs that aren't needed that computers can do faster and better, when instead scientists, young scientists, should be learning the ideas and using a calculator to get these results and not doing it trying to replace the computer. You don't need people to be using these old methods that fail. It's also easy to use. There's an intuitive user interface, and an analyst, if a lab sets its protocols up, can spend seconds working on a particular case, not hours or days. So what's the power of DNA? I'm not going to show any data. I usually show lots of STR peaks. I'm not going to show an electropherogram. I'm just going to talk about the idea for the non-DNA people here. So the idea is we have evidence. Here it contains 3 people standing in for genotypes in a mixed DNA sample, and we want to identify a reference sample. The source of DNA power is being able to separate those genotypes from the mixed samples, and TrueAllele does that separation automatically. You see the different fading of people. That's because data will only give you a genotype up to probability if you extract all of its information to preserve it. And then you end up, say in this example, with 3 separated genotypes. That's done without looking at any reference. It's completely objective. Those 3 separated genotypes can be compared with anyone. But the older methods lose DNA power. The simple allelists, they can't count DNA amounts. They've applied a threshold. The peak heights are gone. And with those thresholds, they can't separate and they cannot identify. So on the left, we see a picture of many DNA alleles from different people. On the right, there's few DNA alleles. And counting up how much DNA is present from a peak height is the same. A way to think of it is you can ask, is 1 gram of heroin the same as 100 kilos? Well, to the old DNA methods, yes. They can't count. They can't distinguish between 10 or 10,000. They don't work quantitatively, and that's why they don't solve problems. The way to restore the power is to make informative genotypes count, to separate and identify so the evidence can be separated, unmixed, and then made with comparisons to identify anyone as being there or not there. This was the first modern case ever done. Famous case, there have been books and TV shows about it. A man was killed outside of Pittsburgh. A police officer was charged with the crime. He'd been living with the estranged wife of the deceased dentist. And the problem was that there are 13 million people in Pennsylvania, and the most the FBI could come up with their old CPI inclusion statistic was a 13,000 statistic. And the concern that the prosecutor had was that was the only evidence he had, and he was going to lose. So instead, we reanalyzed it with TrueAllele. We had another expert use more information as well. And the score went from 13,000 from the FBI's CPI method to 189 billion. That's about 10 million times more with TrueAllele. Same data, but much better data analysis. And the reasons for this are well known, of this DNA mixture failure. In the federal government in the US, the National Institutes of Standards and Technology started doing studies. In 2005, almost 20 years ago, they released a first study that showed with a very simple two-person mixture, most labs called it inconclusive. I think about 70 labs in the study. Of the 30 or so labs that gave a number, their number ranged from tens of thousands, that's 4 zeros after the 1, to hundreds of trillions, that's a 1 followed by 14 zeros. So clearly, this method wasn't working. It was considered inconclusive by most labs, and the match statistics were all wrong, actually. These older methods have a built-in bias in their manual review. You would think you put the DNA into a computer, it gives you a match statistic. If it's a big number, that shows that it's more likely someone's there. If it's a small number, it's more likely that they're not. That isn't how it works with the current methods used in India and elsewhere. First, an analyst must choose their data. Who ever heard in science of someone choosing their data? That's not science. Then a person decides, is there an inclusion or not? You've already reached the conclusion on the data that you've chosen looking at the answer. That's the thresholds method. And then you calculate a statistic. It should be the other way around. It's known that this is a very biased approach and has led to false convictions. In fact, NIST continued with its study, and in 2013, they did a larger study with different mixtures. This was a 3-person mixture, and there were about 100 labs at this time reporting on the results. 70 of them thought that somebody was there who wasn't there. They falsely included them. 70% got the answer completely wrong and might have convicted an innocent person. Only 6 of the labs out of 100 properly excluded. One of them was using TrueAllele. So these allelists have a very high error rate. This result comes from a validation paper published in PLOS ONE with Cybergenetics and the Virginia lab in 2014. We measured error rates. With match statistics over 1,000, TrueAllele had no error. Between 100 and 1,000, the error rate was maybe 0.00001%. But we compared with different threshold methods that Virginia was leaving before they went on to TrueAllele. With the CPI method, with one threshold, their error rate was 6% false inclusions on simple 2 to 3 person mixtures. And with the stochastic threshold method, where you use 2 thresholds that American labs moved to for 10 years, it was supposed to get rid of all false positives, all false hits, with the trade-off that you'd lose 2/3 of the information. The million statistics went down to hundreds. There was still a 2% error rate. So manual review falsely includes innocent people. In fact, it's so bad that these allelist methods with CPI shut down crime labs. In 2015, the Washington, D.C. lab and a Texas lab were both shut down. The reason for it is that allelists don't have any scientific basis. CPI doesn't work. It's supposed to be measuring how much information is present in the evidence. It doesn't. All it does, when you look at the actual data and you study it, which we did and others have done, it's just counting up how many locations, how many loci, or genetic tests an analyst is reporting. Take the number of reported inclusion loci, divide by 2, that's the expected number of zeros. It has nothing to do with identification information. It's not identification science. And that was shown in this paper that we published that showed there's basically no correlation between identification information and these failed CPI numbers. Manual mixture interpretation is inaccurate. It disagrees with the true information. It's subjective. The workflow must introduce human bias, as others have published on. It's widespread. There are millions of case items around the world that have been looked at. It's opaque. People don't really know why the human analysts are making the choices that they're making. It can only include. You can't exclude somebody. You can't exonerate anybody with these inclusion methods. And the result is most of the results are inconclusive and the cases stay open. TrueAllele by comparison, which I designed over 20 years ago, is designed to use all the data. It's accurate. There have been over 42 validation studies, 8 published in peer-reviewed journals. The workflow eliminates people. There is no human bias. There's no choices. All the data is used. It's been reported in 46 states out of 50 in the US. It's used by many crime labs. It's transparent. Our discovery package is 4 gigabytes. We give the papers, the rulings, the science, the methods, the patents, everything's available, and it's neutral. The statistics can exclude innocent people as well as include offenders. The methods are informative. There's always the statistical result. It could be small, it could be large, it could be inclusionary or exclusionary, but the cases are closed because we get information. Here are some interesting cases. I was going to spend a lot of time telling you about them, but I was asked in the interest of time not to, so I won't. Though this was the entertaining part of the talk. Sorry. I'll just tell you about them. This is a case about 10 years ago in Pittsburgh. There was a serial rapist. It was interesting. He was actually a forger. He was found through a forgery crime he had done on a CODIS hit. Turned out he had raped over 10 women. The match statistic from the old CPI method was in the hundreds of thousands. TrueAllele reanalyzed the same data at the prosecutor's request. On the identical data, the match statistic was a quadrillion. He was convicted and sent to prison for a long time based on his crimes, not based on the computer. This was an incest rape case, Lipinski case, which is uninterpretable DNA mixture. You have a father and a daughter whose DNA is mixed together. With the threshold methods that labs use, the answer is, we don't know, it's inconclusive. Against all these 7 items of evidence, you're seeing in the right column the number of zeros in the match statistic. 15 zeros is a quadrillion. Very informative to the computer, closed the case, man's convicted, sent to prison with the current threshold methods, no information at all. Casey Wilson was a serial rapist in New York. He left a pair of gloves that were detected. The lab said there was insufficient genetic information. It was too complex, nothing to report. This case was interesting in that we received the data in the morning, and by the evening we had sent out the preliminary report back to the district attorney's office to present to a grand jury to begin his prosecution. So when you set up your TrueAllele process to be fast, it can be very fast. Ultimately, Mr. Wilson was convicted. This is a case I could give an entire talk on. In fact, I did at a Pro-Mega meeting, or maybe it was an AFS talk. This is a serial rapist in California. This is DNA from 3 crime scenes. There were 36 evidence items plus a zip tie that led to a CODIS hit. These were all 3- and 4-person mixtures. The lab could analyze none of it. They sent it to us before the trial. The right column with the red bar shows 8 hits across these 3 crime scenes to Billy Ray Johnson. All the evidence was informative to TrueAllele. The blue bar across the center is showing how by finding 3 people, the rapist, the victim, and elimination sample, were able to help the prosecutor tell the story of what happened. When the crime lab finished validating the TrueAllele system they bought, at trial they were able to present results alongside us. Their results completely agreed with ours using TrueAllele. There have been a lot of TrueAllele validation studies, 8 published in peer-reviewed journals. They're all on our website, and we can make them available to you. This study from 2011 was comparing the CPI manual method with TrueAllele automation. What the bars are showing vertically is how much information there is. Essentially, summing it up, is when a person could get a result, and they gave a match statistic, on average, TrueAllele would get 1 million times the number on the same data. So whenever a person got a result, TrueAllele got a result. The next paper with the same group in 2013 asked the other question. When the computer got a result, how often did the crime lab get a result? Even though the numbers were lower, less weight of evidence, how often did they fail? Crime labs failed 70% of the time on the identical data. Computers saying the average match statistic is a quadrillion. Human review is saying 70% of the time, nothing, inconclusive. The case isn't closed. This is another paper that we did with Cleveland Lab. And what it basically shows is TrueAllele has been validated and published up to 10 unknown contributors. It doesn't consider 2 or 3 to be difficult, nor 5, nor 7. It just, the computer solves it for you. It's a mixture calculator that will give you answers. This is a listing of the 8 peer-reviewed studies starting in 2009. Lectures about these papers are on our website, or I can give more talks, which I have there. Each one has many different points. They're interesting. You only need one validation paper for the quality assurance guidelines in the US, but every time there's something interesting, we'll write up another paper. Let me move to DNA databases. So in DNA casework, there's a concept that you have evidence from a crime scene, and you have a reference sample, and when you get genotypes from both, you can calculate a match statistic. All a DNA database does is lift that up to a database. So many labs can upload their evidence to an evidence part of a database. They can upload reference samples to the reference part of the database, and then the matching can automatically happen at the database level with the information being sent back to the participating labs in the database so they can solve cold cases. So every study that validates that a method is working, like TrueAllele for casework, since the database is using, the TrueAllele database uses the same math and the same matcher, it also shows that the database works well. We have a number of database examples. This was an exoneration that we did about 3 years ago. In 2011, a man in Houston, Texas, was murdered. DNA was found under the man's fingernails, but the crime lab called it inconclusive. No use was made of the data, and an innocent man, Lydell Grant, was wrongfully convicted and sent to prison for life for a murder he didn't commit. The Innocence Project of Texas, we work with many innocence projects. We've probably worked on over 50 innocence cases. TrueAllele's exonerated over 10 innocent men. Reanalyzed the identical data, and on a 10% component, we got a strong match statistic of around 1 trillion. We showed several things. It was not Mr. Grant. He was excluded statistically. There was somebody else present. And then one of our TrueAllele labs ran a CODIS search. We found the man in Georgia who actually committed the murder, who, when he was found by police, confessed that he had done it, and Lydell Grant was freed from prison. This is a bus crash from South Africa, disaster victim identification. A long story, but I'll keep it short. Essentially, on the left, there were victim remains of people who who died in the crash, over 15 people. At most, one relative came forward. So either there was no relative or one relative. The relative could be a grandparent or a son. And the police gave up on it. They sent it to us saying, here's an impossible case. And a week later, we sent them back the match results. We ran this as an exercise. Imagine you have all the victim remains. You have the kinship problem. The computer has to solve the kinship problem, infer the genotypes, do the matching, and resolve it. This took, after seeing TrueAllele for 2 days, in Chandigarh at a workshop we ran a few months ago, the 20 students were able to use TrueAllele to ask all these questions and set them up, analyzing the data fully in 1 hour. And then over lunch, the computer solved it. In the afternoon, they looked at the results. TrueAllele's really, really automated. Drug counterfeiting. This was a company sent us data from over 100 counterfeit packages. A crime lab using the old failed CPI methods that crime labs use with thresholds said there was no usable information. There was nothing there. Given the same data, we found over 700 DNA connections and 6 profiles that could have been uploaded to CODIS if that was the project, which it wasn't. The lab spent 1.5 years finding nothing. Nothing. 1.5 years. All the TrueAllele computation was done automatically in a week. We worked on this case with the Cleveland laboratory, Dr. Nasir Butt, and on the left, in this talk that he gave, he shows that there were 30 profiles that human review found inconclusive. All of them produced a result in TrueAllele that was informative, using probability genotyping. There were 12 samples they thought were exclusions. They weren't exclusions. Using a powerful computer, TrueAllele, they found positive matches. The human review was inaccurate and uninformative, whereas the computer was informative and accurate. Here's the database validation that I presented. It's on our website. In 2012 at an American Academy of Forensic Sciences meeting, we looked at how well CODIS, or databases like that use allelist, CODIS, small pond, work compared with TrueAllele. I won't go into detail. You can watch the talk. It's only 15 minutes, but almost done and out of time here. The bottom line is that CODIS and these allelist databases must block mixtures from going up to the database. Even the simplest 2- and 3-person mixtures in the study, if they didn't, they had to block 1/3 of them. 1/3 of the evidence could not be used because if it was up there, it would falsely match. You would find innocent people using a database routinely. How many? That's shown in the bottom right. If you didn't block the upload, most mixture profiles are blocked. The evidence never ends up in the US on CODIS. You'd hit, depending on what stringency you used, 5 to 25% of the database finding the wrong person. And every citizen ultimately would become a suspect in a crime. TrueAllele has a secure architecture. It's firewalled with encrypted SSL database. There's no identifying information. Samples are anonymized. There's password protection. There's an SSH encryption for the connections, and TrueAllele provides the support. It would be good if India had a uniform DNA database that was powerful, that could use its reference samples to actually match against evidence, ultimately, and solve crimes. There are 2 TrueAllele database servers that might be used for reference samples. From my understanding, there's going to be one database in NCRB in New Delhi as the national database, and all the CFSL labs will share a common database. Dr. Jain, director of the DFSS, asked, how can India maintain uniformity across all the states? And at Cybergenetics, we discussed it, thought about it, and our answer was, we will give TrueAllele viewer software for free to all the SFSL labs for uploading their reference profiles to CFSL and NCRB. So with that, I would like to conclude, thank people for coming and being here today. For if you don't know already, there's going to be a 2-day workshop on the TrueAllele technology hosted by CDFD in Hyderabad on Monday and Tuesday of next week. If this intrigued your interest and you'd like to attend, please contact Karthik, who's here, from Global Forensics. Here's his phone number on the top right. Thank you for being here, and maybe we'll see you in Hyderabad next week. Thank you very much.